welcome to Jonah's video on CRISPR treatments or solutions for cancer. Now, I'm going to give you a little background on CRISPR and medicine. CRISPR is like many other advances in science and medicine. CRISPR was inspired by nature. In this case, the idea was borrowed from a simple defense mechanism found in microbes, such as a bacteria, to protect themselves against invaders like viruses. These microbes capture snippets of the intruder's DNA and store them away as segments called CRISPR. If the same virus tries to attack again, those DNA segments, turned into short pieces of RNA, can help an enzyme called Cas to find and slice up the invader's DNA. After this defense system was discovered, scientists realized that it had the makings of a versatile gene editing tool. With a handful of years, multiple groups had successfully adapted to the system to edit virtually any section of DNA. First in all the cells of other microbes, then eventually in human cells. I'll give you a background on cancer. Cancer is an overgrowth of cells caused by a certain virus that disables a gene, a cell that prevents it from dying. When it is supposed to be there for the cell, overgrows, causing a tumor. Scientists have been researching CRISPR and have been using it to cure many different types of diseases. If CRISPR technology can find and locate a strand of DNA within cells that have this disabling virus, then remove the strands and replace it with a healthy copy, we can hopefully prevent cancer or slow it down. Ever since scientists realized that changes in DNA have caused cancer, they have been searching for an easy way to correct those things by manipulating DNA, like CRISPR. Although several methods of gene editing have corrected those changes, CRISPR is like many other advances in science and medicine. CRISPR was inspired by nature. In this case, the idea was borrowed from a simple defense mechanism found in microbes, such as a bacteria, to protect themselves against invaders like viruses. These microbes capture snippets of the intruder's DNA and store them away as segments called CRISPR, or clustered regularly. Several methods of gene editing have been developed over the years. None has really fit the bill for quick, easy, cheap technology. But in 2013, when several researchers showed that gene editing tool called CRISPR could alter the DNA of a human cell, like very precise and easy to use pair of scissors. The new tool has been taken to the research world by storm, remarkably shifting the line between possible and impossible. As soon as CRISPR made its way into shelves and freezers of labs around the world, cancer researchers jumped in at the chance to use it. The first trial in the United States to test a CRISPR-made cancer therapy was launched in 2019 at the University of Pennsylvania. The study was funded in part of the NCI testing type of immunotherapy, which patients' own immune cells are genetically modified to better see and kill cancer. CRISPR was used to remove 3 genes, 2 that can interfere with the NY-ESO1 and another that limits the cell's cancer-killing abilities. CRED at National Cancer Institute, the therapy involves making 4 genetic modifications to T cells, immune cells that can kill cancer. First, the addition of a synthetic gene gives T cells a claw-like protein called receptor. In addition of a synthetic gene that gives T cells a claw-like protein that sees NY-ESO1, a molecule on some other cancer cells. Then CRISPR is used to remove these genes, 2 that can interfere with the NY-ESO1 receptor and other limits the cell's cancer-killing abilities. Product of this study is called NYCE, T cells, was grown in large numbers then infused into patients. We had done a prior study of the NY-ESO1 directed T cells and saw some evidence of improved response and low toxicity, said the trials leader. If the study is correct, we should slow down and completely erase cancer using the Cas9 plus T cells of CRISPR. Also, it would be very beneficial technology. That's all I have today for you. I hope everyone has a good day, and thank you for watching.